Now we're ready to discuss proteins. Protein structure can be discussed at 4 different levels. As you probably know, you've seen these different levels in biology, I'm sure. So I'm just going to go over them quickly, but with functional groups in mind, which you probably did not have in biology classes. Okay, so the simplest level is the primary structure of protein. So what that is just basically the sequence of amino acids as shown in this handout. So when the hormone insulin was discovered, the sequence of 51 amino acids was determined, as I'm going to show you right now. This is the sequence. So it turns out there's a chain A and then a chain B, and they're connected together through the cysteine disulfide bonds, which we'll talk about later. But right now the primary sequence is basically, what is the order? What's the first amino acid and what's the second, etc., etc. So there's an analogy between the primary structure of protein and words that the letters of the alphabet make up. So you know that words are formed when the letters are properly sequenced, and that conveys some kind of information. Likewise, proteins are formed from proper sequence of the 20 amino acids. So that proper sequence is needed to make biochemically active protein. So think about how many possibilities are there for the 51 amino acids that's in insulin. It's like, if you do the calculation, 1.5 times 10 to the 66 possibilities, yet our body reliably produces just the right one, which illustrates the precision of life process. All right, so once we know the sequence, the primary level, then we're looking at the secondary level, which is the regularly repeating order special arrangements of amino acids near each other in the protein chain. So these result from the hydrogen bonds, and I want to differentiate the result of a secondary structure versus the tertiary structure. The secondary structure comes because of the amide backbone. So the hydrogen bond that we're talking about here is the result of amide backbone. Excuse me. And not the side chains. All right, not the R group off of the each of the amino acid, but the amide backbone itself. All right, it's a regularly repeating order arrangement. All right, and there are two types that you're familiar with, and one is called alpha helix, and the other one is called beta sheet. I'm sure you've heard of those before. So what the secondary structure is asking is does the amino acids, once you know the order and what amino acids are present, do they exist in straight chain, or is it exist in some other way? Okay, so the alpha helix, which is what is shown here, is a result of so this is the amino acid chain, and notice that I think it's about 3 amino acids away, this carbonyl is close to the hydrogen on the nitrogen of the frame. the backbone, and so it could hydrogen bond. Same thing here, same thing here. So all these hydrogen bonds exist, and this is a ribbon-like structure, and this is more with the models. And if you, this is a side view, and this is looking at it from the top, looking down. It almost looks like a cyclic structure because it's like a spiral staircase. And that is held in that shape due to the hydrogen bonding. So in other words, like if I have a long chain like this, and if I spiral it like this in my finger, right? So that's the form that it exists in, and the way in space. So without my finger, it exists like that. And the reason why it exists like that is because there's a hydrogen bonding in between to hold them together in that spiral shape. Okay. Now beta sheets are it's from like a hydrogen bonding like this. So pretend there's one chain and then another chain, and in between, if you line up correctly, you have alternating hydrogen from the nitrogen. We could hydrogen bond with the carbonyl on another chain. So notice these hydrogen bonds in that, and that is what is responsible for the beta sheet as shown here. So they kind of look like a fan. I made one for you to illustrate. So pretend that in this beta sheet there is a long chain going this way, and it could wrap around and come around like this, or it could be another chain altogether, but they are attracted to each other by the hydrogen bonding between the chains in this way. Okay, so there's a picture of it right here, and this is a ribbon diagram of what a beta sheet would look like, how they're arranged like this. Okay, so these hydrogen bonding between the nitrogen hydrogen, so the NH between that and the carbonyl, that's what's responsible for the hydrogen bonding. It's not that much. It's about 5 kilocalories per mole. It gives you that much, 5 kilocalories per mole more stability by having these hydrogen bonds. But because there's so many of these, that's what causes the shape to stay in that way. All right. The beta sheet is the type that are found in silk, and if you look at I think this is a good analogy of these secondary structure before we go on to the tertiary structure, is that you don't probably remember these phones, but they used to have these cords, and so the primary structure would be a strain out of this, and if you let it spiral around like this, that's like alpha helix type of secondary structure. So, and then if I take this, and then does it exist in a straight chain like this, but in a spiral fashion, or do they fold up like this in a globular fashion? Then that is tertiary structure, which is what we're about to talk about. All right. So the tertiary structure is more like the folding of the various regions together to make a more compact structure, like a sphere or a linear, more stronger structure. So this interaction is caused by the side chain groups. So I want to distinguish that interaction between the side chain groups of amino acids. All right. So there are 4 types of these interactions that make up the tertiary structure. So this actually is a structure of hemoglobin, and notice how they're alpha helixes, and notice how they fold in certain fashion. And the reason why tertiary structure, the 3D shape of a protein is so important is because that determines the activity of the protein. So if you do not have the right shape, then even if the primary and the second structure is correct, it does not do the job. This is especially crucial for enzymes. They have to have the right shape in order to have the correct activity. All right. So let's go over the four different types of interactions that's possible between the side chains of amino acids. So these squiggly lines are indicating the protein peptide chains, and the strongest of these is the disulfide bonds, because that is a covalent bond. In other words, the two electrons are shared between the sulfur and the sulfur of the cysteine, and so because two each are shared and it's a covalent bond, it's a pretty strong bond. So this is a disulfide bond. And that could only occur when we have cysteine amino acids available, just like you saw in the insulin. Notice that the cysteine form disulfide bond here, connecting the chain A to chain B in two places, as well as there's an intra-molecular cysteine disulfide bond causing this to curve around like that. All right, so that's very important. The second strongest interaction, so as we go from left to right, this is the strongest interaction and this is the weakest interaction. So the next one is the ionic attraction, and this happens if we have a side chain with an acidic residue like aspartic acid, and then we have another chain with basic residue in the side chain. They're going to be attracted to each other because one's going to be negatively charged and the other one's going to be positively charged. And this could happen within a chain. So imagine a chain like this, and let's say, well, I have an aspartic acid here and a lysine here. They're going to be attracted to each other. So then notice that the chain will tend to fold like this to be close to each other. All right. The next one is hydrogen bonding among the side chains. So notice, remember in the beginning when you looked at amino acids and how they were classified, you know that there were amino acids called polar amino acids, but not charged, right? So neutral polar. So that's where if we have OHs along with CO1Os, notice how they will hydrogen bond like this. All right, so I'm going to end it here and continue this in the next lecture.